Hi everyone. So this is part 2 of microbiology of soil topic. Okay, so in this video you're going to learn about the different types of microbes that are present in soil. So they are bacteria, fungi, actinomycetes, archaea, and protozoa and algae. All right. So how much of these microbes in soil? So the proportions aerobic, majorly we have aerobic bacteria, about 70% of the total microbes in soil. We have anaerobic and actinomycete, each about 13 to 14%, and we have fungi 2%, and others are about 0.2%. Okay, so in terms of biomass, per tons per hectare, so how much biomass or weight of these bacteria, these microbes in soil? So we have bacteria about 1 to 2 tons per hectare. We have fungi the most, about 2 to 5 tons per hectare. Okay, and the rest is about 0 to 0.5, 0.2 to 2.5 tons per hectare. So every organisms in the soil are important, so they have their own function. So number one, they are the photosynthesizers, photosynthetic bacteria, photosynthetic organism. Of course, plant can convert the solar energy to fix carbon dioxide and change it to their own food. All right. Decomposes, bacteria and fungi, we know that they can decompose waste product, decompose cellulose, can degrade cellulose, and other plant litters. All right. They are also mutualists. So mutualists are a type of interactions between two organisms that will give benefits for both organisms. Okay, we have also pathogens that can cause disease to plant, all right? And then root feeders, those nematodes and microarthropod worms that can actually eat up the plant roots that will cause crop yield loss, all right? Also, we have protozoa, nematodes, and microarthropod as bacterial and fungal feeders. Okay, we have shredders, those that can actually shred the plant litters and degrade it into smaller particles so that other organisms can consume. Okay, and also we have higher level predators. Okay, the nematode, larger arthropod that will eat up all the small organisms. Okay, first we're going to talk about bacteria. So bacteria is the major organisms found in soil. So how much is it per gram? We can count 10 to the power of 10 cells. So that is how much? Billions, trillions maybe of cells per gram. Okay, so of course, if you go down deeper, so you can find anaerobic bacteria, and based on next generation sequencing, the diversity of microbial communities or bacterial communities in soil is about more than 10,000 taxa, so 10,000 different taxa of organism, of bacteria per gram of soil. So that means you can have different families, even for different genus, different families of bacteria per gram soil. Okay, so that high in diversity. All right, so let's go through what are into specific types of bacteria that are present in the soil. Okay, so when we talk about the dominant culturable soil bacteria, so those that can be cultured on in the lab, okay, so we can find Arthrobacter, okay, so it is up to 40% of Arthrobacter are from the total culturable bacteria. It is important for nutrient cycling and biodegradation. We have streptomyces, okay, a type of actinomycete, okay, involved in biodegradation as well as antibiotic production. We have a lot of pseudomonas and also bacillus. Okay, so these four types of organism are easily found in soil. Okay, you can culture them on plates. Okay, so other examples of important autotrophic soil bacteria. So autotrophic are bacteria that can actually make their own food, okay? So other than photosynthetic bacteria, we have also these autotrophic bacteria such as Nitrosomonas, Thiobacillus species. So these are bacteria that is involved in the nitrogen and sulfur cycle. Heterotrophic soil bacteria, so those bacteria that actually lives on that need to convert sugars, okay, need carbon for their growth, okay, eat up other organic materials, okay. So for example, actinomycetes, we have bacillus. So actinomycete is the one that produce the earthy odor when there is raining. You can smell the earthy odor. That is actually the smell of actinomycetes, and also it produces antibiotic. We have bacillus and clostridium, both are spore former but and gram positive, but bacillus are aerobic, clostridium are anaerobic. So they are important for carbon cycling, okay. Bacillus are production of insecticides, okay. So insecticides, for example, Bacillus thuringiensis can actually kill insects that is plant pathogens, okay. And also some bacillus can produce antibiotic. Clostridium, okay, involves in carbon cycling, but also can produce toxin. We have methanotroph, methane oxidizers. We have Caprivedis necator, rhizobium. You heard a lot, I'm sure, about rhizobium. That is important in nitrogen cycles with legumes. Frankia, nitrogen fixes nitrogen with the non-legumes plant, okay. Also as agrobacterium, important plant pathogen that will cause crown disease in plant. Okay, so bacterial profiling. So so nowadays we can actually know the total number of bacteria in a soil, in soil. So previously we as we know, so not all bacteria are culturable, okay. Not all bacteria can be cultured in the lab because of the complexity of the nutrient that is needed. We cannot grow them in the lab, or we don't know how to grow them in the lab. But with the advance in technology of sequencing, we have this next generation sequencing or metagenomic analysis. We can know the different output taxonomic unit of OTU of bacteria in soil sample. So culture dependence mean with culture independence mean bacteria that is identified from the not culturable, okay, from the genomic and metagenomic analysis. So whereas in the culture-based technique, so where we can culture them on the lab, so we can find lesser organisms. So these are the different type of bacteria. We have Acetobacter, a lot of Actinobacter, actinomycetes, okay, and then Bacteroidetes, Firmicutes, and so on. All right. Okay, so that is bacteria. So now we have actinomycetes. So Actinomycetes is actually a type of bacteria. It's a prokaryotic organism classified as bacteria, but it is unique enough to be discussed as an individual group. So that's why we normally, discuss, we mention bacteria and actinomycetes. So we don't actually just silent it. So, okay, so the number of actinomycetes normally